Okay, thank you very much for your nice presentation. I would like also to acknowledge Alfredo, Javier, Fermin for the kind invitation to participate in this outstanding meeting. And what I would like to do in the next few minutes is just to share with you the results of the investigation we have done in the lab for several years, and is related with trying to understand the mechanisms that are triggered by a deficient metabolism of methionine and S-adenosylmethionine, and how these alterations could actually condition or can participate in the progression of liver diseases. So, as you know, methionine is an essential metabolite that is mostly metabolized in the liver, where it can be either used for the protein synthesis, or alternatively it can be catabolized through the so-called one-carbon metabolism, leading to the synthesis of S-adenosylmethionine over here that can lead to the synthesis of polyamine that then could be back to methionine again through the methionine rescue pathway. So some alternatively can be through the many transmethylation reactions can be transformed into S-adenosylmethionine, then, homocysteine, then to homocysteine that can be remethylated back to methionine, in which the methyl group donor is in this case tetrahydrofolate, or can alternatively start the transsulfuration pathway that is the source or one of the sources of glutathione, one of the main reducing agents in the cell. And the decision of whether homocysteine goes back to methionine or enter the transsulfuration pathway, that very much depends on the viability of methionine. If methionine goes down, then transsulfuration is blocked, and then the remethylation pathway is stimulated. And on the contrary, if methionine goes up, then remethylation of homocysteine is blocked, and then homocysteine will go through the transsulfuration pathway. And there is one molecule that is S-adenosylmethionine that basically plays a pivotal role in this decision. So we were very much interested in this, in S-adenosylmethionine, its synthesis, regulation, and so on. So this is the actual molecule that is used as a food supplement currently in many countries. And from the biochemical point of view, it's a kind of interesting one, because of the presence of this sulfonium in the molecule that provides a huge plasticity. So this propylamine group is the one that is used for polyamine synthesis. Probably the most recognized function of S-adenosylmethionine is its capacity to transfer this methyl group to many biological acceptors, including nucleic acids, proteins, small molecules, hormones, and so on. So it's basically the main or the most important alkylating agent in living cells. It participates at least in as many reactions as ATP, so it plays a key role in cell biology. So we were very interested in this molecule because of this observation we made many years ago, in 1988. We found that in the liver of cirrhotic patients there was an important impairment in the production of S-adenosylmethionine. So the actual reaction of synthesis of S-adenosylmethionine is that one here, is in the blue color. Is a two-step reaction. So first the substrates methionine and ATP bind to the active site of the enzyme, that is methionine adenosine transferase, leading to the synthesis of S-adenosylmethionine and tripolyphosphate. And then this tripolyphosphate is hydrolyzed into pyrophosphate and phosphate to promote the release of all the products from this, from the active site of the enzyme. In mammalian cells, there are two different genes coding for 3 different isoforms of methionine adenosine transferase. And the expression of these 2 genes is tissue-specific. So in adult liver, the gene what is expressed is MAT1A, that encodes an alpha-1 catalytic subunit that organizes into homodimers or homotetramers. I would refer in general to the liver enzyme as MAT1-3. I will not make any differentiation between these 2 isoforms. So this is the form that predominates in adult liver. However, in extrahepatic tissues, like fetal liver, and I have to say that also is the case in hepatocellular carcinoma, this gene is not expressed anymore, and the gene expressed is MAT2A, that encodes an alpha-2 catalytic subunits that form homotetramers that can be regulated by the interaction with a regulatory beta subunit that is not related genetically with the catalytic ones. And this, I have to say that these beta subunits, regulatory subunits, do not interact with the liver enzyme. Okay, so MAT has been very well conserved throughout evolution. And actually, the identity at the amino acid level between alpha-1 and alpha-2 subunits is larger than 80%. However, the regulatory and catalytic properties of the liver enzyme and extrahepatic enzyme are pretty much different. So MAT13, the liver enzyme, is stimulated by the substrates of the reaction, methionine and ATP. This has not been so far demonstrated in the case of MAT2A, that in turn is inhibited by the product of the reaction, S-adenosylmethionine. So that means that the liver, having these particular isoforms, is able to metabolize most of the methionine, or at least 50% of the methionine from diet, and also is able to maintain higher S-adenosylmethionine levels than in any other tissue. And this is important to preserve a well-differentiated and functional hepatocytes. So in addition, even though I said that there is a high degree of homology between different MATs all along the evolutive scale, the liver enzyme has an extra regulatory mechanism, and this is due to the presence of this particular residue. This is a cysteine at position 121 that is located in this flexible loop in the structure that is at the entrance of the active site. It should be over here. All the side chains of amino acids in these positions contribute to this active site. And what we observed is the oxidation or nitrosylation of this particular cysteine residue inactivates the enzyme. Okay? So you have to be aware that the SAM synthesis is quite expensive in energetical point of view. The synthesis of a single molecule of S-adenosylmethionine involves the consumption of the 3-phosphate groups of a molecule of ATP. And in some situations, it probably is interesting to have this high-capacity metabolically enzyme downregulated. So this is why we think that the liver enzyme has this particular residue there, providing an extra method for the regulation of its activity. All other MATs so far described have a glycine residue perfectly conserved at this position. So what we think that happens in the liver is that MAT can be in two different situations in equilibrium. So under normal conditions, MAT would be reduced and is perfectly active. They would maintain normal SAM levels, high levels of S-adenosylmethionine, and then the hepatocyte will be happy, functional, very well differentiated. However, under stress conditions, either because of deficient synthesis of glutathione or increased production of reactive oxygen species or nitric oxide, then this equilibrium will be pushed towards the oxidized form of the enzyme. Cysteine 110-1 will be oxidized or nitrosylated. Then the activity of the enzyme will be impaired, leading to decreased S-adenosylmethionine content. And this is nice if there is a transient situation just to avoid the energization of the cell under stress conditions, for instance. But if the situation chronifies, then what we know is the hepatocytes lose their function and start proliferating. So we have different data using different experimental models actually demonstrating this schema here. And this could also explain our former observation of impaired S-adenosylmethionine in the liver of cirrhotic patients where there is actually oxidative stress. So we thought that this is actually one of the factors that may decondition the progress of liver diseases. But if we want to demonstrate that, or in order to demonstrate that, we obtain a knockout model, mice, where we were able to knock out the expression of MAT1A. However, the expression of the gene MAT2A is perfectly normal, was not affected, so meaning that all the embryogenesis, the liver would be with no alteration, the synthesis of SAM would be ensured by the expression of the gene MAT2A, and also the S-adenosylmethionine supply in extrahepatic tissues would be normal. So if there is any alteration, because, this gene, MAT1A, starts expressing just after birth. So if there is any alteration in the liver associated with a deficient S-adenosylmethionine production, would be just after birth. So this is just to show you that effectively, we were able to knock out the expression of the gene MAT1A, but in adult liver, in these knockouts, the expression of the fetal gene MAT2A is maintained, perhaps as a compensatory mechanism, because otherwise the liver would not be viable. So it needs somehow to have the capacity to produce S-adenosylmethionine. But because of the regulatory properties of this particular form, there is not the same capacity to metabolize methionine, and in consequence, we see accumulation of methionine in blood. You see important hypermethionemia in the knockout. So just to summarize the features of this of our knockout, there is the MAT activity was only 30% as compared to the wild-type glutamates, and likely this activity is contributing by the expression of MAT2A. That leads to a 75% reduction on S-adenosylmethionine content in the knockout liver, hypermethionemia, and also a defect in the synthesis of glutathione, so suggesting that there is some oxidative stress in there. So the main question here is that these particular mice with no hepatic methionine adenosyl transferase developed any disease. So by the age of 3 months, the liver looks pretty normal, but it was more sensitive to liver damage processes, either induced by choline and methionine deficient diet or induced by the administration of carbon tetrachloride. However, if we just wait for a little bit, 8 months, by the age of 8 months, they develop non-alcoholic steatohepatitis, as evidenced in here. You see all these fat accumulations and lymphocyte infiltrate inflammation in the liver parenchyma. And also by the age of 15 or 18 months, all of them, all the mice, all the knockout mice, 100% of them develop hepatocellular carcinoma. So our model actually was deficient in the production of S-adenosylmethionine in the liver, and in fact, this seems to be a factor that contributes to the progression of this disease. So we have a disease that progresses on time, somehow resembling what happened in humans, and we were very much interested in understanding the mechanism of all these pathogenic pathways. And to do that, we use a proteomic approach. I mean, this was the schema. We wanted to compare the proteome of wild type and knockout livers at different time points, 1 day, 15 days, 3 months, and 8 months after birth. Because this is probably all these alterations, thinking that all these alterations were the early events leading to the development of hepatocarcinogenesis. So we use a two-dimensional genetic trophoresis approach. So proteins are separated. We got something like similar to all these spot patterns, basically 1,500 spots, both for the wild type and knockout. We were able to compare these maps, leading to the identification of differential spots that could be identified as particular proteins by using different mass spectrometry approaches. So we did all these experiments, all these comparisons, and we end up with more than 100 differential proteins that are represented in this heat map over here. So each column is one of the time points we studied, and in the rows correspond to the different proteins identified. So we were interested in some proteins that are were altered very early after birth, 1 day after birth. There are many biological processes that were involved in this pathogenic process, but the main one were metabolism, mitochondrial function, and oxidative stress. So we didn't study in depth this metabolic alteration, just to give you an overview of the magnitude for the metabolic adaptation associated with the deficient S-adenosylmethionine synthesis. You see that there is, amino acid deficiency, glucolysis, hydrocarbons, lipids, so on. Okay, there were also a mitochondrial impairment, as was further confirmed. This is a deficient inner mitochondrial potential, and this deficiency was mediated by the alteration of particular proteins that were detected in our proteomic experiment and were further confirmed using Western blot approaches. So you see here the increased accumulation of ATPase beta chain as soon as 1 day after birth. Just remember that there was no phenotype, there was no pathology in the liver at that time, but we were able to see these early markers, the accumulation of ATPase beta chain that was paralleled by the increase of the expression of the corresponding gene. A protein that actually attracted our attention at that time was prohibitin. You see, we identified in our proteomic analysis a deficiency on prohibitin that was further confirmed 1 day after birth and 8 months after birth, as you can see here by Western blot. There was not a lot of data about the function of this protein when we identified the prohibitin. We knew basically that it was located in the nucleus of the cell and also in the mitochondria. The nucleus was regulating the activity of PRB and E2F, and in the mitochondria it was somehow stabilizing the respiratory complexes. So if there is no prohibitin, then these complexes would be destabilized and could be digested by mitochondrial proteases, and this seems to be the case, as we proved with two cytochrome C oxidase subunits. We're able to see also the decrease of these two proteins, probably as a consequence of prohibitin deficiency. I have to say that these alterations on prohibitin and cytochrome C oxidase subunits occurs at the protein level, but not at the RNA level, as you can see here. So the expression of the gene is normal, and the regulation occurs at the protein level. We will never identify these alterations using genomic approach. So at that time, we wanted to know if prohibitin was actually a primary target of S-adenosine methionine. And to understand that, we moved to an in vitro system. These were primary hepatocytes, and we inhibit using a typical classical inhibitor of methionine adenosine transferase. So we expose the cells to cycloleucine, this inhibitor. So there's no activity of methionine adenosine transferase. Nothing happened 12 hours after the incubation, but 24 hours later, the addition of cycloleucine to the culture media, you see that there was an important downregulation of prohibitin. So the difference here probably is related with the half-life of the protein. So another way to do the same thing, that probably more physiological, is to remove methionine from the media, avoid the use of any chemical inhibitor of MAT. So we just remove the methionine from the media, leading to the downregulation of prohibitin 24 hours later. And interestingly, if 12 hours after methionine deprivation we add to the culture media S-adenosine methionine, we were able to prevent the drop on the amount of this protein, of prohibitin. And finally, to just end this part of the talk, just to test if this was a feature that was specific of our experimental model, we tested another one that is sort of obese mice. You can see there is also decrease on prohibitin and cytochrome C oxidase 1 or 2, and also there is a decrease of prohibitin and COX-1 in obese patients, conditions that are likely at risk of NASH development. So basically what we think is that deficient production of S-adenosine methionine can lead to a PHB downregulation, mitochondrial impairment, oxidative stress, metabolic dysfunction, and then to the onset of a liver disease. So PHB, prohibitin, is playing a pivotal role in all these pathogenic mechanisms. So then we wanted to understand which were the cellular functions that could be regulated by prohibitin. So we used in this case human hepatoma cells, PLC cells. What we did was to silence prohibitin. In all of our experiments, the prohibitin levels dropped more than 80% and was accompanied by the downregulation of a functionally related protein, PHB2. And the downregulation of PHB led to the impairment of the capacity of hepatoma cells to grow. The proliferation was decreased as we test by two independent methods. This is a colorimetric method. The silence PHB deficient cells have the open circles here, and you can see that they grow much more slowly. In addition, the silencing of prohibitin also led to the loss of the capacity of hepatoma cells to grow in anchorage-free media in soft agar, as you can see here. So silenced cells die from anoikis. So the deficiency of prohibitin triggered apoptosis. It was evidenced at 12 hours later, the silencing procedure, and it's a time-dependent increased reaction. It is paralleled by the cleavage of PARP, and it's a process that is dependent on the activation of caspase 7 and downstream caspase 12. And it's not dependent on the activation of caspase 3 and caspase 9. So meaning that this apoptotic reaction is mediated or is associated with endoplasmic reticulum stress and not by mitochondrial dysfunction. We made some transcriptomic analysis. We got something like more than 400 differential genes upon silencing of prohibitin. I'm going to show you just a couple of networks. This one with some morphological features or cellular morphology alterations over here, and here suggesting that there was an impairment on NF-kappa B function. In fact, we were able to demonstrate by Western blot that there was an activation by the downregulation of I-kappa B alpha, and also to the increased translocation to the nucleon of NF-kappa B P50. In this other network, there are several things I want to comment. This part one here is related with alteration of the regulation of PRB and E2F pathways. This is something that was already known, so somehow we're telling us that our selection of genes and our bioinformatic analysis is good. This is what somehow expected. There are some factors that are important. Some of them are here. This one, DDIT3, or this called also CHOP, is activated by ER stress. We could confirm the upregulation of this factor by Western blot. So upregulation of this leads to the upregulation of TRP3, and this one is an inhibitor of AKT, and therefore we can see also impairment of MAP kinase pathway as evidenced by different target genes. In fact, we could measure hypophosphorylation of ERK and also upstream kinase MEK. And in addition, we also observe downregulation of cyclin B, evidencing cell cycle arrest. We also made some proteomics on that. I'm not going to get into details of all this table, but there were some proteins that for us were very important. One of them is insulin-like growth factor binding protein 1, because there is a connection between prohibitin and one of the most important survival pathways in the liver, that were mediated by the IGF action. Another one is LN2, that a deficient of LN2 tell us that probably there is a uncontrolled release of calcium from the endoplasmic reticulum leading to the apoptotic response, or could be one of the factors involved in the apoptotic response we have observed upon prohibiting silencing. And there were another two proteins that are stathmin, and in the table this other one, this proteasome activator complex unit 2, that are suggesting a dysfunction in the proteasome. So also could be confirmed by the accumulation using this panel here in PHB deficient cells of ubiquitinating protein. So what we think is that there is some the deficient of prohibitin in the liver cell leads to endoplasmic reticulum stress, apoptosis, and also parallel with impaired proteasome function. Just in the last few slides, I would like just to summarize some very preliminary results on a knockout that was kindly provided by Dr. O'Malley. It's a knockout deficient on PHB. We used heterozygotes because the null mice was not viable at all. So we saw that the knockout mice is much more sensitive to choline and methionine deficient diet. This is the result we got in this, using these liver sections after 3 days of this diet. And it's also, and the recovery response is also impaired in the knockout liver. So we fed the animals for 20 days with a choline and methionine deficient diet, and then we analyzed the liver 7 days later after a normal diet. So the body weight was more or less restored in the wild type, but this is not the case in the knockout, and you can see that the huge difference in the liver parenchyma here. Part of these differences probably are due or mediated by the defect in AMP kinase. So we have done also some transcriptomic, although this is very preliminary, but just to comment that we saw an upregulation of IL-1 and also TNF, so suggesting that somehow there was some pro-inflammatory phenotype in these mice that was further confirmed just by looking at the accumulation of some pro-inflammatory cytokines in the blood, such as IL-12 or TNF-alpha receptor 1 or 2. And this was further supported by the increased sensitivity to treatment with pro-inflammatory insult as LPS. So you see that the survival of the knockouts were severely reduced. So our feeling is that the hepatocytes are not quite happy with half of the prohibitin, but that the lymphocytes are not, leading to this pro-inflammatory condition. It has been recently published a paper in pathology of a knockout that is deficient on prohibitin, specifically in the liver, and it develops cancer in a few months, and all the animals die. So prohibitin likely is playing an important role in the hepatocarcinogenesis. So just to summarize, I hope to show you evidences to support that the adenosine methionine deficiency is an important factor involved in the progression of liver diseases, and part of all the mechanisms at least are mediated by a defect on prohibitin 1 through the interaction and the regulation of many important cellular processes. And that's all. Thank you very much for your attention. Oh, sorry, the most important slide is that one here, just to thank all the people that has been involved in all these studies. So thank you very much for your attention.